Hi guys, today we're looking at AQA A-level biology, the structure of DNA and RNA. So let's get into the video. Don't forget to subscribe. Now, DNA first of all stands for deoxyribonucleic acid. Now this stores the genetic information, which is the instructions that codes for the proteins that make up an organism. The proteins in our body, for example, the keratin that makes up our hair and nails. Now DNA contains deoxyribose sugar, and that's a pentose sugar, so it's got 5 carbons within it. Now there are 4 bases within DNA. There are adenine, thymine, guanine, and cytosine. And RNA is much smaller than DNA. RNA stands for ribonucleic acid. RNA transfers that genetic information from DNA to the ribosome so that proteins can be synthesized, such as the keratin we mentioned earlier, or things like enzymes. Now, RNA is made up of ribose sugar, so DNA deoxyribose, RNA ribose. Now, there are 4 bases within RNA, and they're a little bit different to DNA. So, adenine, uracil, guanine, and cytosine. And you'll notice the key difference here is that RNA contains uracil rather than thymine. Now, ribosomes, they're the site of protein synthesis. So to put it simply, they're where proteins are made. Interestingly, they're made of protein and RNA, and they're used to make proteins. DNA and RNA are basically long chains of nucleotides. DNA and RNA are the polymers, and the nucleotides are the monomers, which is fascinating. Now, a nucleotide is a monomer for the DNA and RNA polymers, as I've just mentioned. And nucleotides are made up of 3 things. Number 1, a pentose sugar, which has got 5 carbons within it. Number 2, a nitrogen-containing organic base. And number 3, a phosphate group. Now, for number 1, the pentose sugar having 5 carbons, it's easy to remember this as pentagons have 5 sides, and pentose sugars have 5 carbons, and I always say this to my students to help them remember it. At the bottom, I've got a little nucleotide here, and we can see the phosphate there. We've got the pentose sugar. So, for example, a nucleotide in DNA will have deoxyribose sugar, whereas a nucleotide in RNA will have ribose sugar. And then finally, we have the nitrogen-containing base, the organic base. And if you want to know about the other biological molecules, check out my previous videos on carbohydrates, lipids, and proteins. So, if we join these nucleotides together, we'll see that they have phosphodiester bonds between them. And the phosphodiester bond is on the right-hand side of the screen here, and we can see 2 red lines showing the ester bonds, and the circle showing the phosphate. And it's phosphodiester because there are 2 ester bonds. Just like carbon dioxide has 2 oxygens, a phosphodiester bond has 2 ester bonds. And I know this sounds tricky to begin with, guys, but it gets a lot easier the more you practice it. So well done for watching this video. When a condensation reaction forms a bond between the phosphate group, shown by the circle on the right-hand side, of one nucleotide, and the sugar of another, this forms phosphodiester bonds. So we've got a nucleotide at the top, and we've got a nucleotide at the bottom, and we've got a phosphodiester bond between them. Now, phosphodiester is where the phosphate group has 2 ester bonds, as I mentioned a little bit earlier. But with a word like phosphodiester, it really does take a bit of repetition to get it in your head. Now, a chain of sugar and phosphate molecules makes up the sugar-phosphate backbone of DNA. And we can see here the sugar-phosphate backbone going all the way down. Okay, the sugar is the pentagon, and the phosphate is the circle. Now, when many nucleotides bind together in this manner, this forms a polynucleotide. So think about maths. A polygon is a many-sided shape. A polymer is made up of many monomers. Okay, monomer meaning one, polymer meaning many monomers. And a polynucleotide is simply made up of many nucleotides. Now, next of all, let's look at the DNA double helix. And I've got a somewhat complicated diagram on the right-hand side here, but let's simplify it. So DNA is made of 2 polynucleotide strands that coil around each other. And we can see we've got 1 strand on the left, okay, and we can see the phosphates there, with our little sugars there. And don't worry if this looks a bit complicated at the moment, it will make much more sense as we go through. And on the right-hand side, with thymine at the top, we've got our other polynucleotide strand. And remember, it is sugar, phosphate base, sugar, phosphate base, all the way down. Now, this is known as the double helix because it coils around, okay? And I'll show you an image of that in a moment. And within this double helix, we have complementary base pairing. Now, you should have already looked at enzymes, where enzymes are complementary to substrates. Well, in this case, we have bases that are complementary to each other. So adenine is complementary to thymine, meaning adenine always forms hydrogen bonds with thymine. Guanine is complementary to cytosine, meaning guanine always forms hydrogen bonds with cytosine. And between the adenine and thymine, we have 2 hydrogen bonds. And between the guanine and cytosine, we have 3 hydrogen bonds. So let's have another little look at the diagram on the right-hand side. Well, at the top, we can see adenine and thymine. And we've got 2 dotted lines there between those nucleotides representing the 2 hydrogen bonds. Now, if we look at the bottom of this diagram, we can see we've got guanine and cytosine, and between them, we've got 3 dotted lines representing 3 hydrogen bonds. So that's what we mean where adenine always binds to thymine and it has 2 hydrogen bonds, guanine always binds to cytosine and it has 3 hydrogen bonds. Okay, next of all then, DNA is anti-parallel. Again, another tricky concept to get your head round. Now, obviously, if something's parallel, it runs directly next to something else, okay? They run horizontally or vertically next to each other, okay? Now, anti-parallel is where you flip one of them on its head and they run in opposite directions, but they're still kind of parallel to each other. So let me show you this as an example. Now, at the top left of the diagram, we've got our phosphate group, okay? And we represented that as a circle when we looked at the nucleotide. Now, at the bottom right of the other strand, we've got that 5' end, that phosphate group, okay? And that would have been represented as a circle as well. So you can see that they've been flipped over on their heads, so they're running anti-parallel. Now, don't worry about that too much until we look at DNA replication together, but I just want to introduce it now. Now, the double helix structure of DNA was discovered by Watson and Crick in 1953, but they did use a lot of work from other scientists, such as Rosalind Franklin, who took that famous X-ray diffraction picture. So yes, they did discover it in 1953, but they had a little bit of help. Now, in contrast to all of this, RNA is much simpler. It's shorter, it's usually single-stranded, and it's still a polynucleotide, but it's smaller and it's single. Now, here's another diagram showing the DNA double helix, and this one's a little bit nicer now. So we can see that it's coiled up, forming the helix. And I always say to my students, if you've got a helix piercing, that's the coiled bit of the ear pierced. Well, here we've got the double helix because we've got 2 polynucleotides coiled around each other in a helical manner, in a twisted shape. And we can see there we've got our lovely adenine and thymine nucleotides or bases joined together, and we've got our guanine and cytosine bases joined together as well. Now let's recap everything, because I know I've hit you with a lot of new content here, a lot of quite complex terms and concepts. So to put it simply, DNA and RNA are polymers of nucleotides. Nucleotides are made up of a sugar, a phosphate, and a base. Condensation reactions join the nucleotides together by forming phosphodiester bonds. DNA is a long, double-stranded polynucleotide with a double helix structure, and adenine is complementary to thymine, and guanine is complementary to cytosine. And next we have adenine to thymine with 2 H+ bonds, or hydrogen bonds, and guanine to cytosine with 3 H+ bonds, or hydrogen bonds. And finally, the strands in DNA are anti-parallel, meaning they run in opposite directions to each other. So well done for watching this video as part of your revision or study. That is a fantastic step towards getting the best grades you can possibly achieve. Don't forget to like, comment and subscribe, and I will see you in the next one.